Okay, so since all eukaryotes have mitochondria, but not all have chloroplasts, what can that tell you about the order in which these organelles evolved? It tells you that mitochondria must have come first, right? What's the evidence for this? Well, the existence of endosymbiotic relationships now suggests that if they can happen now in a very tolerant environment, then they probably could have happened a long time ago in a very extreme environment. Similarity of chloroplasts and mitochondria to specific bacteria, especially in size. They are double membrane and similar biochemical properties to a lot of the bacterial plasma membranes. They have replicative processes resembling binary fission during mitosis. They contain their own genome. That is the big one. They contain their own genome in a circular DNA, just like prokaryotes. They have the machinery to make tRNAs, ribosomes, and other equivalents to transcription and translation of DNA. And their ribosomes resemble prokaryotic ribosomes very, very much. So there's a lot of similarities between prokaryotes and these mitochondria and chloroplasts that suggest that endosymbiosis is a very good theory. So really, when we talk about eukaryotes, they are basically just this mesh of prokaryotic parts. All right, you have the mitochondria, you have plastids, which include the chloroplasts, you have the nuclear genome, right? And it's also been suggested that parts of the mitochondrial genome have actually inserted themselves into the nuclear genome, so that we know that there probably was some sort of gene transfer. But the key here, just like they were doing with the prokaryotes, is looking at that small subunit ribosomal RNA, or the SSU rRNA, because it's present in all organisms, which means that it formulates the deepest level of ancestry that you can possibly have. So the closest prokaryotic relatives to mitochondria are the alpha proteobacteria, which is a relative of the cyanobacteria. They have their own DNA, but you have to remember that they are now interdependent, meaning that these mitochondria and chloroplasts probably once were able to survive on their own with their own DNA and their own ribosomes, but now they are no longer self-sufficient, which means that they have their own DNA, but they are genetically not able to be free-living anymore. Some are coded in organelles, some from nuclear, and some are a mixture of both. So the DNA then, as I told you before, is probably a mixture of nuclear DNA from the larger host and then DNA from the mitochondria and chloroplasts. So when we look at, this is talking about primary endosymbiosis, meaning the first symbiotic event. There is such a thing called secondary endosymbiosis, which is right here. Okay, so this is how they think that cyanobacteria and algae diversify so well, because they engulf different types of plastids. All right, plastids are going to be all of the pigments that can harness the power of the sunlight. We classify them as plastids. You'll hear me talk about chloroplasts mostly, but you'll see when we talk about red, brown, and green algae that there are different types of pigmentation that will give you different colors. All right, so secondary, tertiary, all of the other endosymbiosis, other than primary endosymbiosis events, are going to give you all of these multiple membrane envelopes, the nuclear envelope, the double membrane in the chloroplast and the mitochondria, things like that, which suggests that this isn't something that just happened all of a sudden. It's probably going to be endosymbiosis occurs over very long time. So plastids then are acquired several times by secondary endosymbiosis. The primary endosymbiosis would be the prokaryote intaking another prokaryote. Okay, so that would be right here. Secondary endosymbiosis then means that if you already have a prokaryote with a prokaryote inside, well, we call that a eukaryote. So now it's a eukaryote that is intaking more prokaryotes here. Okay? So each endosymbiotic event will then add another membrane to it. Okay, here. So then you can count the membranes, and you kind of can see that how many times those endosymbiotic events occurred. Now, because systematics is not a exact science, all right? We have been constantly remodeling the 3-domain system, all right? We went from a 5-kingdom, now we have 3 domains, now we have protists in 20 plus kingdoms. It's constantly being revised. But the big question that everyone wants to argue about is, how did these 3 domains arise? Was it a serial traditional hypothesis like this, where there's a sequence, or is it, that the division, the evolutionary lines are skewed, they're blurred by multiple, division events that occurred? So we use this traditional vertical line, right? That's how we like to do it. You've seen clades, we always use them in vertical lines. But eukaryotes evolve horizontally here, meaning that there's a fusion of all these lineages, all right? The tree has gene transfer horizontally. Now there is not a single common ancestor, it's a community of several ancestors that were very promiscuous, you might say, and had a lot of gene transfer, meaning that they transferred a lot of characteristics. So this increased complexity makes variation extremely possible, because now you've opened up a lot of events that can occur to diversify these 3 domains. So in the case of eukaryotes, here you see this huge, eruption of diversification, of evolution, because of the emergence of eukaryotes. First with structural diversity, then with metabolic diversity, then they become multicellular. Now we have this hypothetical phylogenetic tree that looks something like this. Okay, with your ancestral eukaryote somewhere down here. Okay, because we like to do this in a linear fashion, all right? So you have your multitude of protists here, and then the line is blurred where plants start to form, then fungi, and then animalia here. So, should it be just plants, fungi, and animalia in kingdoms, or should they all be in sub-kingdoms? I mean, this is this is a daunting project to take on, to try and do this kind of cleanup and try to organize all of the organisms that we can. So should they be somewhere else? I mean, the protists, they can fit into specific categories of plants, fungi, and animalia, but then since they have so many characteristics of all 3, it would be hard to, have them be split into 2 different kingdoms, even if they're the same. Even if they are the same organism. So we're going to look at a little bit of protistan diversity. I'm going to give you the highlights of what you need to know for your test. All right, so I've already made lists of all the things. Now, we're going to follow this little tree. So the first one that comes off is going to be your Diplomonadida and your Parabasala. All right, these, you will be able to recognize them if you know that they lack mitochondria. Okay, they are here.